So I mean, that's pretty much how to train your own AI algorithm on IKOSA. You need to understand about annotations, you need to understand about regions of interest, but then it's just that very simple point and click workflow. Wait 15, 20 minutes and your app's trained. But of course, that's only half the story. So you can train apps to your heart's content, but they're no good if you don't use them to actually do some analysis. So we saw how to do some analysis earlier with a pre-trained application that we've made available on the platform, and to do it with to analyze with one that you've trained, you need to what we call deploy the app, and that rounder will change to blue. As you can see, there was already one that was deployed down the bottom here before, but yours has now been deployed. Which means you could submit image analysis jobs. So we select image analysis over here. The first tab we see are the pre-canned or pre-trained applications that we have made available. Just wait a second while that data loads. And on the other tab, you'll see the application that we just deployed from the webinar demo training. So we select that as the app we wish to use. Then we have our image library from this project. We don't want to use ones that have annotations because obviously we use them in training. So we would use a different image that has no annotations. So we can select webinar sample 5. And then what you can also do is within that image, if you've drawn a region of interest with no annotations in there, and you can see this one has one region of interest, then you could tick this box and just analyze that region of interest. You don't have to analyze the whole slide. So this will really speed up your analysis work if there's only a certain part of that slide you're interested in. You can ignore everything you're not interested in. So you don't have to let the AI look at the whole slide, draw an empty region of interest, and simply analyze that region of interest. It sells. That's all we had, and we start the job. And so then, as before, we showed the job. It will be under we've got the pop-up here to say confirm it's been submitted. The job will be on the analysis tab, as we showed before. It'll be in the active tab. This one will take a little longer because it was a more complex image. Here we have the one we just submitted a few seconds ago, and here we have a few that we finished earlier. And then the output of that analysis job, again, you have the image with the visualization. So you can easily see everything that the AI has found. So this one was a whole image. Can't see a lot there, but if we open that up in a new browser tab, so we can really zoom in, you'll see that on the left of the image we have the original image that was uploaded to the platform. On the right hand side we have all of the cells. Again, it's maybe a little bit far away, but we have all of the cells that the algorithm found. As you can see, it's done a pretty good job of actually finding all those cells. And then in the middle we have the same thing, but there are numbers assigned to those cells, and those numbers basically correlate with what's in the CSV file. So if we have a look at the one we submitted, the webinar sample 5, which only had the region of interest. So here is just the region of interest that we wish to analyze. It's much easier to see here the numbers that are assigned to everything that it found. So all of these cells is what that application found within that region of interest. And then we have the CSV file associated with that, which gives you, and this is just a preview that we have. There's a summary of that cell, so it's giving you a total number of objects found in that image, 116,000. It's giving you some other summary statistics, but then we also have a CSV file which is available for the actual cell itself. So we can download that. It's 60 meg, so it's going to take a while. You can preview quite a lot of them here in the browser. Unfortunately, this one is quite large, so it's a little bit big for the browser. So we would need to download that file. Here, we'll download that one, and then when we open it in Excel, which will take a second, you'll see the full information about every single cell that was found. So there's unique ID for each one, but then we've got the ratio, the perimeter, the circularity, eccentricity, kind of everything you might want to know about each of those individual cells that the AI algorithm found. And you would take this information and you could apply as much further data analysis as you wished to really solve your use case. And yeah, to be fair, that's really the IKOSA platform. That's how to train your own bespoke AI algorithm. That's how to analyze images. Yeah, that's the heart of it. It's all browser-based, point and click, flexible, ready to solve your use case.